Hi everyone, my name is Casey and today I'm going to be discussing the liver, including its structure and function, and some related medical tests and pathophysiology. So let's get started. As you can see in the diagram here, the liver is located in the upper right portion of the abdomen, just below the lungs and under the diaphragm. It is wedged, almost triangle shaped, has two lobes, and weighs approximately 3 pounds. Blood flows from capillaries in the GI tract, spleen, gallbladder, and pancreas to the liver via the hepatic portal vein. The liver receives oxygen-rich blood from the hepatic artery. The diagram also shows in green here the common hepatic duct, which connects the liver to the gallbladder, and the common bile duct, which connects the gallbladder to the small intestine. If you look at the diagram here, the pinkish rectangle-shaped blobs are hepatocytes. Hepatocytes are the functional unit of the liver, meaning that they carry out the liver's multiple and diverse functions. The hepatocytes are organized into lobules, which are represented here, and there are actually around 1,000 of these lobules in the liver. Each lobule has vasculature to allow the hepatocytes to exchange materials with the blood. And each lobule connects to ducts, which together form the common hepatic duct. I'll discuss the common hepatic duct more in detail when I talk about bile secretion. The liver plays a critical role in metabolism, nutrient balance, waste secretion, and more. Some of its important functions include processing nutrients from food, sugar storage, production and secretion of bile, which helps with metabolism, production of cholesterol, which is secreted into the blood and bile, removal of toxins, processing and storage of vitamins, maintenance of fat, amino acid, and glucose levels in the blood, and synthesis of proteins. The liver has evolved over time to take on these diverse functions and is fairly conserved across species. As mentioned before, the liver receives nutrient and toxin-rich blood from the hepatic portal vein, which carries blood from capillaries in the GI tract, gallbladder, spleen, and pancreas. By filtering toxins and waste from the blood, the liver prevents these materials from traveling systematically and damaging other tissues and organs like the heart. For example, the liver removes potentially dangerous bacteria from the blood, and in doing so, plays a part in our immune function. Once harmful materials are filtered out of the blood, the liver breaks them down and secretes the products into the blood or into the bile. These materials can then be processed by the kidneys and are excreted in the urine or travel through the digestive tract and are eliminated in feces. To aid in digestion, the liver secretes a substance known as bile, which contains bile salts, bicarbonate, cholesterol, phospholipids, bile pigments, and organic waste. To review what we saw on the first slide, the bile is first secreted into small ducts that come together to form the common hepatic duct. The gallbladder is a small sac that branches off from the common hepatic duct and stores bile in between meals. During a meal, the gallbladder is stimulated to contract, and bile is pushed into the common bile duct and into the small intestine, where it can aid in digestion. The bile salts in the bile work to solubilize dietary fats, thereby increasing the rate at which the fats are digested and absorbed. Bicarbonate in the blood, on the other hand, helps to neutralize stomach acid in the small intestine and prevent further damage. As mentioned before, organic waste in the bile travel through the digestive tract and are eliminated from the body. The processing of carbohydrates and fats is a main source of energy for the body. The liver works to convert carbohydrates and fats into glycogen, lipoproteins, and triglycerides. The structures of these can be seen down here. Glycogen is stored in the liver until it is converted into glucose, and this conversion is necessary for the body to have enough glucose to use for energy. Most fat, on the other hand, is packaged into aggregates with protein that are known as lipoproteins. Lipoproteins and triglycerides are secreted into the blood and travel to adipose tissues where they are stored. Amino acids cannot be stored in the same way that carbohydrates and fats can. The liver is responsible for synthesizing proteins from dietary amino acids, which can then enter the blood and travel systematically. Amino acids can also be metabolized for energy. and this is done through the production of acetyl-CoA, which is fuel for the citric acid cycle, and production of NADH and FADH2, which can be used in generating ATP through the electron transport chain. The process of amino acid catabolism also produces ketone bodies. Ketone bodies are an important source of energy for the brain when glucose levels are low. Finally, the liver helps with disposal of nitrogenous waste generated during protein degradation through the process of urea metabolism. One medical test that checks for liver pathophysiology is a liver biopsy. During this procedure, a long needle is inserted into the patient's abdomen and a few liver cells are collected. The cells are then checked under a microscope for signs of scarring. A liver biopsy may be done if a tumor-like mass is seen on imaging tests, an abnormal liver test can't be explained, to determine severity of a liver disease, to determine effectiveness of an ongoing treatment, or to monitor a liver after transplant surgery. A biopsy can help to diagnose or stage hepatitis B or C, autoimmune hepatitis, alcoholic liver disease, hemochromatosis, and Wilson disease. We'll actually talk about Wilson disease in a few slides. Liver function blood tests assess levels of enzymes and proteins in the blood. The purpose is to see if the liver's exocrine or metabolic functions have been affected. It is administered using the same process as a normal blood draw, and the blood is analyzed using laboratory experiments. These liver function blood tests are useful in screening for liver infections, monitoring disease progression, measuring disease severity, monitoring side effects of medication. Alanine transminase, aspartate transminase, and alkaline phosphatase are common liver enzymes, and low levels of these can mean that the liver cannot carry out its metabolic functions. ALT helps to convert proteins into energy. AST metabolizes amino acids, and ASP is important for the breakdown of protein. The total protein, albumin, cholesterol, and glucose levels are important to monitor to make sure that the liver is secreting these materials into the blood so they can be used around the body. Finally, bilirubin is produced during the breakdown of red blood cells. Low levels of bilirubin may indicate that the liver cannot properly break down organic waste from the body. One organic material that the liver is responsible for processing and eliminating is copper. Wilson disease is an autosomal recessive disorder that results in an accumulation of copper in hepatocytes and leads to liver and brain tissue damage. It is caused by a mutation of the ATP7B protein in the liver. Under normal circumstances, dietary copper travels to the liver through the blood, where ATP7B either binds it to apoceruloplasm or packages it into vesicles to be secreted into the bile through exocytosis. The copper bound to apoceruloplasm is eliminated from the body in urine, and copper in the bile is eliminated in feces. Mutant ATP7B remains unfolded and is degraded by the endoplasmic reticulum control system. This results in an accumulation of free copper in hepatocytes, which damages their membranes and allows free copper to travel in the blood and become deposited in tissues like the brain. Free copper in the liver also leads to free radical generation in the presence of hydrogen peroxide. And these free radicals can also travel around the body and cause damage in other tissues. Some common symptoms of Wilson's disease are liver failure, impaired movement, which occurs when the basal ganglion is damaged, dementia, which occurs when copper and free radicals accumulate in the cerebral cortex, Kaiser-Fleischer rings, which are seen here and are caused when copper is deposited in the cornea, and increased levels of copper in the urine. Children with Wilson's disease tend to present more with liver failure, and older adults with Wilson disease tend to present more with neurological symptoms. Some hypothesize that this could be caused by different mutants of the ATP7B protein, but more research is necessary to reach a conclusion on this. A liver biopsy and a liver function blood test can both be used to diagnose and stage Wilson disease, by measuring the liver's exocrine function and level of tissue damage. To review what we went over today, the liver plays a critical role in metabolism, nutrient balance, waste elimination, and more. 2 tests to check for liver pathophysiology are liver biopsy and a liver function blood test. One example of a liver pathophysiology is Wilson's disease, which is caused by improper excretion of copper due to a mutation in the ATP7B protein. That's all for today. Thank you.